Hi everyone, welcome to our Hot Topics in Science webinar, bringing together leading researchers from two European funded projects, BOOSTER and HelEx. My name is Pierre Compère and I'm the moderator of this webinar. BOOSTER and HelEx projects are two Horizon Europe funded projects. Both aim to adapt crops and crop systems facing climate change. We'll discuss cutting-edge advances in plant sciences, genome regulation, pollinator monitoring, and AI-driven environmental assessment. HelEx and BOOSTER mobilize researchers from several European countries. Let's start with BOOSTER. Increasing drought conditions are threatening yield stability, particularly in staple crops. BOOSTER aims to solve this by exploiting natural genetic variability to make crops tolerant to drought without sacrificing yield. BOOSTER partners are focusing on three specific species. First, maize, a vital cash crop for food and fuel, but one that is highly sensitive to water stress. Then, teff, an African superfood that is naturally tougher than maize, but still requires irrigation. And last but not least, lavagrass. This is our secret weapon in a way. It's a wild relative of teff that acts as a resurrection plant capable of surviving even after losing 95% of its water. The goal is to understand what makes lavagrass so tough and transfer that capability to maize and teff while helping to create a more favorable environment for production in such harsh conditions. So how will BOOSTER achieve this? By employing a dual strategy. First, on the genetic side, researchers are identifying the specific DNA sequences in lavagrass and teff that regulate drought response and transferring these features into maize to improve its resilience. Second, they are developing natural biostimulants. They are using extracts from brown algae, seaweed, and beneficial microbes found in drought-resistant soils. These act as eco-friendly boosters to help the plants cope with stresses. By integrating these methods, they aim to reduce yield losses and strengthen the bioeconomy in both Europe and Africa. Let's move on to our second project, HelEx. Started in 2023, HelEx tackles a problem that has been growing since the 90s, the impact of extreme heat and drought on European agriculture. The sunflower is part of the solution. It is naturally robust, but we need to push its limit further. HelEx aims to provide breeding companies and farmers with new varieties that are not just drought tolerant, but also resistant to heat and high soil salinity. Our objective is to ensure that sunflowers can continue to thrive in the changing European climate, securing production for food and biofuel industries. The strategy behind HelEx is to go back to the source. Researchers are tapping into the genetic reservoir of wild sunflowers originating from North America. These wild plants have evolved in harsh ecosystems and hold the key genes for survival. They plan to integrate these wild genes into cultivated European sunflowers. The focus isn't just on survival, it's on a triple output. First, oil production, maintaining economic yields. Two, the quality, of course, keeping protein level high for nutrition purposes and non-food uses. And three, ecosystem services. This is crucial. HelEx will ensure these new varieties still produce enough pollen and nectar to support honeybees and wild pollinators. Now you have the context. It's time to discuss and to discover some results. This webinar is divided in 3 sections, and within each section, 5-minute 5-minute presentation will showcase breakthroughs that are shaping the future of crop resilience, biodiversity monitoring, and climate-smart agriculture. You'll be able to share comments and ask questions by writing throughout this webinar, and thanks to the dedicated tab. And I'll be your spokesperson in order to share your comments and questions to the panelists. So it's time to start with the step one. Let's talk about drought regulation at a genome level, and I'll give the floor to Thomas Hartwig, who's a group leader at Max Planck Institute for Plant Breeding Research from Germany. Thomas, are you with us? Can you step on the stage, please?
Yes, I am, if you can hear me.
I can hear you, and I'll give you the ability to share your screen. Thank you for being with us.
Great, and thank you, Pierre, for the great introduction. So let me introduce you into one of the work packages of Booster and how we're trying to improve the drought tolerance in maize and teff. This work package has three major goals. The first one, primarily we're trying to get high resolution maps of the genomes of these two species and where potential transcription factor binding sites are. These are the major regulators of gene expression. We want to do this across a diverse population of maize and teff so that we can have a better understanding where natural variation at these regulatory sites function. And we want to particularly identify and potentially change the sites that are important in response to drought conditions. To do this, as already mentioned, there are two major crops that we are focusing on here, which is maize as one of the top breeding crops, and then teff because of its desiccation resilience. There is multiple molecular assays that we are performing. We're going to look at the expression of genes in response to drought and also across the different varieties. There's also methylation differences that we want to understand, so how the epigenetic changes may affect or are different between the ecotypes. And then also there is a new assay that I'm going to mention in a second, which is called MOA-seq, which looks at the genome responses. One of the key things that we are doing here is that we're also looking at hybrids. So instead of just comparing many different genotypes, we're particularly interested in comparing them with each other, and we can do that in hybrids, looking at different genotypes within single plants. We do this then so we make a population of very diverse hybrids, and we can compare them under different conditions, for example, under drought, and we can compare we can then use, for example, these different molecular approaches, such as, for example, this MOA assay that allows us to see where the transcription factors are binding, and we can use the genetic or epigenetic diversity between these different genotypes in the plant to see if one or the other site was bound, for example, more, if the transcription factor preferred to bind to the mother or the father site. This is how these experiments then look like. So we grew, this is just the maize part, but we did similar things for teff and for lovegrass, where essentially we would grow these plants to a very similar genome developmental stage. And then we would expose them to water withholding or drought conditions and harvest different parts of the plants that are severely usually affected by these drought conditions. So this is then essentially a summary. So we make these populations that we are able to compare if a transcription factor genome-wide prefers one side or the other, and we can compare this across this entire hybrid population. We can better understand where there is genetic differences and where there are also, for example, differences under specific conditions. When we did this, we found over 200,000 sites across this maize population. This is shown here in blue across the different maize chromosomes, where all these different binding sites occur. Some of them were already sites that we are known are phenotype or causing phenotype differences. And we can compare this with additional phenotype data that comes, for example, from genome-wide association studies or from selection or domestication differences, and then see where there are differences. And we can then do this, for example, at local levels, so compare it at specific genes. If we see differences, for example, in the binding of a transcription factor, in the promoter, for example, here, but we also can do this over long distances in the genome to see if one site affects another site that is very far away. And so we can do this under normal and drought conditions, and then get these potential differences of where there are potential regulatory differences. But once we have these sites, now the really crucial part is that we need to validate them. We need to find out if they really have a function in drought. And then that brings me to our next section, which is then the second work package.
The second work package that is led by Hilde Nelson. Hilde, you are from Belgium. You're group leader at the VIB Gen Center for Plant System Biology. So you're going to talk about a multiplex genome editing approach to modulate cis-regulatory elements, isn't it? Hilde, are you with us? 1, 2, 3. Otherwise, it's going to be hard for me to share your results. Hilde, please be with us. Or Thomas, are you still here?
Yes, I am. I'm still here.
So you might need to help me on this.
No worries. I'll do my very best. So as I said, and as I introduced, a really key part is that we need to validate these. And one of the ways how we approach this or try to validate this is with this second work package. And the main goals here are to really comprehensively understand or compare maize and teff. So these assays I just shown to you, where we can see differences. But the interesting thing for us was to better understand how maize and this desiccation tolerant teff, as well as lovegrass, would actually then differ, right? Where there are genomic and gene differences, we need to validate them because these sites, because all we had shown in work package 1 are that they're existing, but whether or not they are related to drought and how they potentially function, we do not really understand at this point, and we need to validate. And then we also need to see if there's ways to do this in teff. So both in maize and in teff, this was a crucial aims of this work package.
I think Hilde was maybe.
Hilde's back. Perfect.
Hilde, I've seen you in a...
Sorry, I was kicked out.
That's all right.
Yeah.
So we're listening to you. So Thomas shared with us the work package 2 goals. So you can now focus on the results.
Okay, very good. Thank you. So to achieve those goals, we have several tasks to deliver. And first of all, we will go also into the more drought-tolerant Nindensis species and also do exactly the same there as what Thomas described for the 2 other species. Once we have the data for the 3 different species, we will do a comparative genomics approach to look what are the drought responsive elements that are specific for each of the 3 species, but also the ones that are in common. So we get a better understanding how the drought responsiveness is regulated in the 3 different species. Once we have all this information, we will start to do a functional validation of these allele-specific MOA-seq elements or cis-regulatory elements that we identified. And we will do this both in maize and in teff. In maize, there is already a little bit more genetic tools available, so that's why this is a little bit more elaborate than what we will do in teff. But basically, it would boil down to the same thing. The first thing we would do is make loss of function of alleles of the key drought responsive genes. This is an approach that most of us know. We call this making mutants. We knock it out. The second approach is really to look at the modulation specifically of these binding QTLs or cis-regulatory elements, and then see what happens and also how it relates to knocking out the key responsive genes. And finally, we will also use available data on phenotypes in different lines already to see which of the alleles that we identified would be the best ones. And finally, all the lines that were generated will be tested under drought assays. As said, the similar approach will be taken for TEF. And once we have all this data, we will put it together, and we will be able to formulate an advice on how the future genome editing approaches, for instance, can be taken in order to achieve better drought tolerance in either of the 3 species. And in the next slide, I will just highlight some of the technical approaches that we have been taking in maize to modulate these transcription factor binding sites or cis-regulatory elements that were identified already in work package 1, what Thomas just explained. So the output of this data in a more concrete way is actually that we see that certain occupancy is different in drought stress than in the well-watered stress. And this regulatory sequence, for simplicity here, is indicated upstream of a gene body, but sometimes this can also be downstream of the gene body. And so once we have these positions where the differences occur, then we can come up with genome editing strategies in order to start to modulate these binding sites or cis-regulatory elements. And we have 2 approaches to do this, and this is exemplified in the next slide. The first one would be to excise these cis-regulatory elements in the background where we see them. So Thomas explained that he did the MOA-seq in different inbred lines. This is now an example of a more susceptible line like B73 and a more drought-tolerant line. And for the first gene, you see that there is a clear difference in occupancy at this peak. So you see the red peak is much higher than the blue peak in the Ohio 43, which is the more drought-tolerant. So the approach that we would take for this gene is try to cut out specifically this cis-regulatory elements. We do this by designing 2 guide RNAs flanking such a peak, and we choose these guide RNAs as such that they are conserved in both the tolerant and the more sensitive species. Once we introduce this in both species, we would aim to remove this cis-regulatory element in both of those inbred lines, and then we will see whether this affects the transcriptional regulation of the downstream genes upon drought. And of course, the ultimate dream would be that simultaneously this would also give us a better drought tolerance or a different response to drought. As you can see from the scheme, we will not just do this for 1 gene, but we have selected several genes, and we will do this in a multiplex way. So for the moment, we are on our way to generate the different lines, and then we can analyze them. So in the next slide, we can then see a different approach to do this, where we will start to insert the different cis-regulatory elements. And this is also a genome editing approach, but it is a little bit different in context. There we will use the CRISPR-Cas9 system to make a double-strand break. And by choosing the guide RNAs also in the accessible chromatin region, so in the promoter region, so to say. And once the cut is made, we also provide the plant cells with an enhancer element, which we now call an enhancer element, but it can be, for instance, a drought responsive element that we select from, for instance, Nindensis or TEF. And this one would then be inserted using non-homologous recombination into the cut site that was created by CRISPR-Cas9. Once this enhancer is there, this would then stimulate the gene expression of the downstream genes, which we can then monitor again either by qPCR and later on by phenotypic analysis. This technology is not so widely used, and for this we really had to test many different ways to get this fragment actually into the exact position. We now succeeded in doing it and now are fine-tuning this looking at the sizes of the enhancer element that we can use for this, and also improve the efficiency and the multiplexing of the fact that we can do this for multiple genes simultaneously. And so this then concludes my part of the work package, but I hope you can see that this was only part of a lot of work that has been done. And for this, I want to thank all the partners of work package 2.
With the mic on, it's better. Thank you, Hilde. Thank you, Thomas. I have 2 questions for you, actually, just before heading to giving the floor to the next panelist. You're quite enthusiastic about future results, and I understand that you want to copy, in a way, and introduce regulatory elements to the maize genome. But do you think that will be enough, in a way, to copy those regulatory elements, or do you think we will need to change the genes themselves?
Yeah, by changing the genes, you mean upregulating or mutating the genes?
Yes, or maybe like changing the genes, the sequences itself.
Yeah, I think it's going to be a multi-layered aspect. So I think there will definitely be genes where we can make a change in the gene body itself, so that, for instance, a protein has a higher activity or a lower activity. This I can also imagine, and that's definitely possible. But for other genes, we have seen that transcriptional regulation is an important way to start regulating this. So I think if we need to really think towards the future, it would be a combination by making edits into the regulatory sequences, but also combined with, in another gene, it can be the gene body that needs to be edited. So that still is quite a challenge ahead of us.
Thomas, I'm sure you agree with that.
I absolutely do. And I think the one aspect that we're also exploring here is we're trying to make changes that at least have a chance of having less side effects. So there's no question that is, as Hilde said, that a combinational approach is probably what will be the most prudent. But we also try to avoid that if we make changes to genes that are maybe in many other pathways, that we have severe side effects.
Okay, and a quick question: what would be your time horizon in terms of transferable results to the private sector? Are we talking in years, I would say, or decades?
Well, I think what we are doing here is innovative. So we need to have some validation, and that's basically also what is happening. But I think once we have this and we know exactly how to interpret this kind of data, I think the incorporation into the private sector could actually go quite fast.
Okay.
And into applications. But maybe I'm too optimistic. But
Thomas, are you as optimistic as Hilde?
I am. I think that the approach is designed to be quite flexible. I think that transformation into different lines is still a challenge and not possible for all maize lines, for example. But if that's not the issue to transferring this information, then the hope is that we can learn, and then the same approach could be done also to commercial lines and then improve them directly. But at the moment, we're still a bit on the learning phases, which of these elements are really key. But I am just as optimistic as her.
Okay, cool. Thank you. Thank you very much to both of you. It's now time to move on downstream, closer to the field in a way and commercial situation. Let's talk about seed resilience enhancement, and let's welcome Dr. Zeroun Tadele, who's a group leader at the Institute of Plant Sciences at the University of Bern in Switzerland. Hi, Zeroun. You'll talk about enhancing abiotic stress tolerance of major food crops using priming agents, aren't you?
Yes. Thank you very much for the invitation. As you can see in this slide, my sponsor is SERI, Swiss educational and research innovation, in addition to the EU, but financial support comes from SERI. Can you change the slide? Move the slide. Yeah. Okay. Our work package 3 focus on development and characterization of new seaweed and microbial biostimulants for improving maize and teff drought tolerance. As you can see below, we are organized under 5 tasks. These tasks are summarized in the following picture. So as you can see, soils, we start from collecting soils from drought areas in Africa, from South Africa and Ethiopia, and Europe, from Spain, France, and Italy. And then, as you can see, you can follow the arrow up. Experiments, we do experiment on these soils collected from different places under drought and the normal water condition. And then at the same time, you can go horizontal. You see, you isolate microorganisms from these soils. And these microorganisms, first we identify them in diversity. And also we check whether they are pathogen or not. We need to isolate, and we work only on non-pathogenic microorganisms. And then we do on non-pathogenic and abundant ones, we do experiment again on maize and teff. This is done by different groups in different continents. And then we identify the best performing strains. And finally, we give this to work package 4. And then work package 4, with the test in the field for maize in Europe, for teff in Ethiopia. So once we do this, we also follow with the mode of action, like whole genome sequence, root colonization, meta-transcriptome. We do this in the meantime. Then I can see also below, we work on the seaweed extracts, which we get from BioAtlantis, different seaweed extracts. Either they are for seed priming or for leaf priming. And then we take this and we do experiment in different places for maize in Bulgaria, for teff in Switzerland. So once we get the best performing ones, again, we transfer the data, the information to work package 4 for field testing, respectively, maize in Europe and teff in Ethiopia. So for this part also, we do mode of action work on transcriptome and the metabolomics. So this is a summary of the 5 tasks we do. And then regarding the soil collection from drought areas in Europe, the collections were made from 14 locations, and the total isolate were about 1,000. And then final study was made on 203, because they need to screen for different purposes, and they need to also check if they have some relevance on drought tolerant. From Ethiopia, from 12 locations, over 300 collections were made. In South Africa, about 150 collections were made. These are strains. And then in Ethiopia and South Africa, they test using polyethylene glycol for the drought response. And then they select about 24% in Ethiopia and 15% in South Africa. So they promote this, and finally 58 strains were shipped to Switzerland. So this is where experiments were made. So regarding the microbial candidate in maize, for those collected from Europe, those enriched in water deficit soils were mostly from phylum Actinomycota, as you can see on the right picture below. So this is enriched in different soil types. That's why this was selected. So the candidate genera are also, you can see 3 of them. So these were promoted to the next step for sequencing and further testing. Can you move? Yeah, regarding the candidate from teff in Ethiopia and Ragnosin indensis from South Africa, also from these 58 strains isolated, the phylum, which dominant phylum were like, you see 3 of them, Pseudomonadata, Bacillota, and Actinomycota. So the genus are also shown in a percentage of their dominance. So drought experiments were made in some soils collected from these areas, shows the following result. At the left side, you can see the soils collected from Ethiopia, from 4 locations, Abala, Lemtena, Dera, and Humbo. And then they test also from these 4 regions, they made from 3 collections, 3 regions, in each location, in each region, 3 locations. So like Eragrosis pilosa field, teff field, and uncultivated field. From this, you can see the teff field was the best because teff is the crop, where uncultivated and the Eragrosis pilosa is just a wild species of teff. So the roots performance were much lower compared to the one from teff field. And then also Dera is providing a very good condition for teff growing in this condition. But while Humbo was worst in this case. At the right side is it shows the performance of teff on soil collected from South Africa, and then that is based upon the chlorophyll content. And then you can see also some strains like strain number 7, strain number 11, and the strain number 18 were found to be the good candidates in this case. Okay, then with this, I move to the seaweed performance under drought condition. The left top panel shows maize. And then you can focus on maybe on the first panel only. FV354 makes a very good response to. Can you go back? Yeah. On seaweed, yeah, good. Can you go a bit down? Yes, no. On the drought, where they says a drought. Okay, I cannot. Yeah. So under drought, normally yes, that lane, yes. So FV354 is seriously affected by the drought. But when you add priming agent seaweed, it's really enhanced the performance of the plant, and then you get a very good seed weight. On the right side, you see the teff performance. You see the under well-watered and the drought condition, and then you can see the figure below where the seaweed 2018, for example, gave significantly higher grain yield for teff. So based upon compared to the no priming and water priming. So this shows that the seaweed also has a very good effect on teff and maize. And then regarding our timeline, we have for these 5 tasks, we are almost on time, except the teff part, because this lesson also we have to learn in the future. When we make a proposal, we have to consider all things, what will happen to us. As you can see now, we are in month 30. We finish our project at month 48. For some, we have some delays, but these delays are being compensated now. We have 6 months delay for 2 tasks because of teff, because the problem is the shipping of soil from one place to another was not allowed. So we need to look at the regulatory condition of each country, and also export permit from Africa to Europe, or also took longer paperwork. So these type of things were affecting us, but now we are almost in the right track. Next. So in work package 3, we have, as you can see, 8 partners. I don't need to mention them, but we are working together in a very good condition, and they are from Europe, from Africa, and the like. Thank you very much.
Thank you, Jeroen. Let's move on directly and to Sujit and Nerakal. Sujit, your head of plant research at BioAtlantis in Ireland, and with you, we're going to talk about field-scale validation of biostimulants.
I hope you can hear me, Per. You can hear me.
Okay.
Yeah. Thank you. Yeah. So in the work package 4 of Booster, we are just extending the work that has been done in work package 3, where we have identified the best biostimulants which is of seaweed origin as well as of microbial origin. So the idea is to see in work package 4 in 2026 in Booster whether how this identified biostimulants will work in a field condition. So before going to the topic, I would like to introduce myself. I am Dr. Sujit Nirakal. I am representing a company called BioAtlantis. We are actively involved in the Booster project. We are an Irish biotechnology company. We entered the biostimulant market in 2024 with stress mitigation as a central strategy. We are grown by a 30% CAGR from that time. We focus both on crops and animals. I will not go into the details of that, but I would like to tell that there is a portfolio of proven biostimulants that we have developed from seaweed extracts and from other botanical extracts. And the details of this, you can go more into the BioAtlantis website and you will get more about that. So next slide, please. So before going to the topic, I would like to just introduce what is biostimulants, because some of the people will get confused biostimulants and fertilizers. So according to the EU regulation, the biostimulants, the definition is the plant biostimulants means the product stimulating plant nutritional process independently of the product nutrient content. This is very important because it is not we can't claim any nutrient content in the biostimulant that is giving the effect. So biostimulants have some unique compounds that is stimulating the plant, and because of this stimulation, you're getting different effects in the plant. For example, the different effects are nutrient use efficiency, tolerance to abiotic stress, quality traits, and availability of confined nutrients in the soil. The last one is for the microbial type of biostimulants. So these are the effects you will get when you stimulate the plant using biostimulants. So these are the objectives what we have using the biostimulants in work package 4. So we have a maize in Europe that we know that because of the climate change, there is a 20% reduction that is going to happen for the maize yield. And also for teff, you know that in Ethiopia, it's a major teff producing country, and teff is being described as a healthy crop. And yet the nutritional average is only 1.7 to 1.8 tons per hectare is the yield in Ethiopia. So we are trying to improve the efficiency of maize and teff using biostimulants. So in work package 4, in Europe, we have different zones that has been distributed for the testing in the field condition for maize using biostimulants. And also for the teff field trials, we are going into the 4 Ethiopian drought-prone teff production areas in Ethiopia, and we'll check in the field condition. So these are the different tasks what we have in work package 4. And the first 3 tasks is based on the production and upscaling of your biostimulants, which has been identified the best. And then we'll go into the different field trials in 3 different zones, in the Mediterranean zone of Europe, in the South East European zone of Europe, as well as the Ethiopian zone for teff. So this is the idea what we have in work package 4. Go to the next slide. So in task 4.1, you can see that we checked about 16 different seaweed formulations we have tested. Out of that, 13 is for seed priming. That means we use it as a seed coating. And 3 for foliar priming. That means we will apply as a foliar application. And from that, we identified 2018 and 2025 as the best for seed priming for teff and maize. And then foliar priming, we have 2022 as the best formulation. So this formulation will be now upscaled. This is the upscaling process is ongoing. And this upscaled formulation will be used for different trials in different zones, what I described before. Go to the next slide. So you can see here, this is the upscale formulation effect, what we are trying to check whether the effect is still there. So you can see that this is the formulation 2025, which has been upscaled, which has been performing the best in work package 3. So you can see that compared to the control, the coated seeds with maize seeds with this formulation has a better germination as well as primary root length improvement significantly. And also you can see that the shoot emergence is higher in by coating this maize seeds with this biostimulant. So this is one of the effects what we have seen. And again, in the case of teff also, we check the same conditions. This is a seed coating in teff that has been performed. And microscopically, when you see the seed germination, you can see that there is an improved germination happening because of in teff because of this seaweed formulation application. And the germination count has been improved. And at a specific concentration, you can see that the improved germination of about 83% attaining with teff seed with the help of this coating. In task 4.2, the same way what we did in seaweed, we will use microbial biostimulants here. So we develop and upscale the biostimulants for microbial formulations. And this microbial formulations will be used as a seed coating agent, and this will be tested for different field trials in different zones for teff as well as for the maize. Yeah, go to the next slide. You can see here, this is the task which is very important actually, because before going to the field trial, all the partners as get together, and then we are trying to see the different designs, what we need for seeing the effect in the field condition. Because what we're trying to see is a 20% or a maximum is 25% difference in yield improvement and other production parameter improvement using the biostimulants. So we need a very strong trial protocol where we have a maximum number of replicates to capture this effect. And also there should be up to the GEP standard, we have to design this trials, and then we'll go further. So this is task 4.4, where we are trying to see it in different zones of Mediterranean zone of Europe, in Pau Plain in Crea, South France in Afebao by Afebao, and southern Spain by Afebao. These are the field trials we'll be doing there in that zone. And there are different trial conditions we are checking there. I won't go into the details of that. And you can see that different parameters what we're going to check there. Next slide, please. This is again the task 4.5. We were testing the biostimulants in the southeastern European zone. This will be done by CPSBB from Bulgaria and Afebao from Belgium. So the Plodif, they'll be checking in Plodif in Bulgaria by CPSBB and Hungary by Afebao. And again, the same type of different field conditions will be checked there. This is the tough field trials we'll be checking. This will be checked in Ethiopia, and we'll be selecting 4 Ethiopian drought-prone tough production areas. And we'll be checking different conditions using the testing both seaweed extract as well as microbial biostimulants. And also the replicates and the importance of different field parameters will be checked. And I think that is the last slide I have. And this is the future what we're going to test. We'll be testing in different zones of Europe as well as in Africa. And we'll test the efficacy of these biostimulants, how this will perform in a field condition. And this will be quite challenging, but it will be an interesting task for us in Booster. Thank you.
Thank you, Sajid. I have the same question as before for the first session in a way. What would be your time horizon in terms of transferable results into the fields?
Yeah, that's an interesting question, because what we'll be trying in the next year is a field trial in the different zones. And this will be a replicated field trials using different cultivars of maize. And once this is, and also in teff also, the same condition. Once this has been proven, the idea is to publish the scientific data and get these formulations to be optimized further in by the companies, and then commercialize it. Because before commercialization, you should also get an approval, the EU approval of CE marking and etc. So it takes about 2-3 years of that. And I think after this 2-3 years of this approval, everything, and then I think we'll be in the market. Yeah.
Okay.
Maybe if I can add also on that point, is now already we are regarding teff and Ethiopia, now a lot of materials are already available there. So once we know, they know the materials, which is a very good one, then they start already the work in the field, in the research station and also in farmers field. So I think in 1 month already they start the experiment. So then what we do is we try to promote this to farmers through like field days and different ways of organizing different events where farmers can come and see the work.
But we'll expect that. Thank you very much to both of you. It's now time to move on to our last chapter of our journey. Let's talk about environmental impact assessments with a focus on biodiversity on sunflower and on the HelEx program. Let's welcome Ralph Wilén. Ralph, you're head of the Institute for Biosafety Research in Plant Biotechnology at the Julius Kühn Institute in Germany. And you are using artificial intelligence to analyze agricultural environmental impacts, aren't you?
Yes, I try to. So because, hello everybody. So we were trying to, because, well, AI comes up as a potential high versatile tool. And so we thought about whether we could use this to tackle some of our problems for the environmental impact assessment in an initial step. And this I would like to introduce right now. Perhaps you could go on for the next items within this transparency and the slide. Go ahead, go ahead, go ahead. Okay, so, and one more, please. So thanks for this slide. And so what we like to anticipate are the trends in sunflower cropping in Europe. And on the right-hand side, you see a map of the different cultivation regions. The darker green, the more is the productivity of sunflowers in this area. And the arrows that are indicating the initial assessment, what could be the development of sunflower cultivation in Europe. And you see, for instance, in the southern part or in some parts of France, we expect that there may not be a big change. For Spain, we have the idea that there may be a decay due to drought events. And for the light green areas in Germany and the western or in the northern part of France, as well as in Italy, we would expect that there could be an increase in sunflower production within the next years. And the tendency, for instance, in the Balkan region, it's a bit shaky, so they are also highly productive. And but we are not quite sure what will be the direction in this sense. So this is, on the one hand, we would like to anticipate what could be the trends. And connected to the trends, there may be shifts in the rotation regimes, and with the shifts in the rotation regimes, so switching, for instance, from rapeseed or from maize to sunflower cultivation to some extent, may also cause a shift in the environmental impacts of the whole agricultural system in this area. So that was the idea. And so, sorry, could you go back, please? Previous slide, please. Yeah. So what have been the methodological challenges? So it is quite easy to get some statistical data of crop production and areas of crops throughout Europe on a national scale. But if you have a look to these regions as indicated here, so that's not really covering the whole country. So there are sub-regions within the nations, and that needs to be identified. And this is much more challenging than just visiting the statistical pages of the European communities or of the FAO. And also the details about cropping regimes within these regions, they are not readily available in scientific data or in publications. So you have to find them up. So we call it gray literature. So it may be recommendations from agricultural services, it may be internet or something like that. And in addition, there may be drivers that bring the one region in front of the others, and may trigger the decision of the farmers what to grow in this region. And so that would also be interesting to find out what are stakeholders thinking about the future of sunflower cultivation in these areas. So to get into or to dive into that, we were employing artificial intelligence system, large language models, and the databases behind it, but has been tested with Claude, Gemini, and Grok. And so these three, there are much more. We haven't tested them in detail. For the trial and error, it turns out that a dialogue prompting I was using was the most promising result, because you could react on the output of the AI system, and you could refine the question or the prompting according to the response that you get. So it's much more a dialogue process. You also could write down a one-pager of prompt where you ask all the questions, all the details that come to your mind, but it's much more laborious. So the whole thing had been performed within the first quarter of 2025. And it turns out that at this stage, Claude was providing a most focused response to the question. Grok, you get also some nice response, but very wordy. And at this time, Gemini was not that you get the details to that extent that you would like to have. So it was actually too general. For all, it holds true that errors occurred, so it was necessary to make a cross-check either by visiting certain web pages and something like that, so that you could compare the outcome of the AI system. But nevertheless, it's not the way that you could say everything was erroneous or everything was on fault. So it has been certain data, certain links to services or something like that that you need to cross-check. And by that, we were able to identify the regions, the main cropping. Sorry, could you stay with the other slide, please? We were able to identify the regions, and but nevertheless, it came across that regional statistics are still rare. So it's not easy to get the regional statistics. And what I performed in the Q4, just preparing this talk here, we were tracking the advancement of the AI models. So there have been new versions of the large language models and data tools that are provided by the different systems. And it was a real fast step or big step forward. But it turns out that, well, if you asking the large language models at different time points, you have to admit that the answers are not exactly reproducible. You get responses, and they have extended the databases, so you also get some detailed data, but sometimes the numbers are a bit shifting. And it turns out, for instance, with the Claude system, that in the survey in the beginning of the year, Claude was focusing on European statistical data, and in the later survey, they are focusing or also using FAO data, so which are not exact in the numbers, and there could be shifts between 10%. So nevertheless, it came out that we could get some crop statistics and trends based on these FAO and AU data that were also used by the large language models. We get to the data of fertilizer use and pesticide use due on recommendations of agricultural services, but on the national scale at this point. We get an idea about water demand and weediness of the sunflower. And comparing the sunflower cultivation data with the data for maize, wheat, and rapeseed, you could easily identify that there may be an environmental benefit by introducing sunflowers into the crop rotations. It was also possible to get some ideas about the expectations of the stakeholders, which brings us back to the regional scale. And so this is what we would like to employ further on. So we used these artificial intelligence systems at the end for a strategy refinement. And so we are now intending to conduct interviews with different stakeholders. The next slide, please, which is summarizing what we will do now in the second part of the project. We use the AI system to found our targeted stakeholder interviews for the next step. We selected some focal regions where we expect some dynamics or some typical dynamics in the sunflower crop production. Southern Spain, as I already said, drought impact, and on the cultivation regimes, central France, the dynamics between the shifts of sunflower cultivation and oilseed crops. In Germany, we have a low level of production of sunflower, but steadily increasing. We have a 200% increase in the last years, even before the Ukraine war. And so it may go to some direction. Actually, it's not clear which kind of crop will be replaced within the rotation regimes. And in the southern France, Romania, and Bulgaria, which are the main production areas, we will try to find out what will be the effect in this region. So we hope by this strategy we will get a trend of sunflower cropping systems and impacts in the different regions, much more detailed than on the national level, and to get hold on dynamic regions or the dynamics, typical dynamics of sunflower cultivation. And together with the work packages that are dealing with economics, we will also get some insight into the drivers for farmers' decision in these different regions. And I think with that, I'm through. And thank you very much.
Thank you. Let's move on to Ilya now. Ilya, you're a researcher and outreach officer at the Carinthia University of Applied Sciences in Austria. And with you, we land on our last country, our last European country, and we're going to talk about DNA metabarcoding for faster and more reliable pollinator monitoring.
Yeah, thank you so much. I hope you can hear me well. Yeah, as Pierre mentioned, my name is Ilya. I come to you from the Carinthian University of Applied Sciences. We're a HelEx partner, and our task is pollinator monitoring with new technologies, new methods. And the key approach we use is genetic approaches, so DNA barcoding. We can already go to the first slide. I'll give you a brief overview. So the project idea and the question behind it was, of course, the study object, the sunflower, which is in Austria not that relevant, but it becomes more relevant, as I've also pointed out. And we try to figure out new approaches to do pollinator sampling in sunflower fields, because traditional methods of biodiversity assessment can be very time consuming, prone to inaccuracies, and especially in Austria, it's very, very difficult to get permits for insect samplings. Molecular methods have already been proven to be an alternative approach to comprehensive biodiversity assessments, although they do absolutely have their limitations, and the approach is key. So we discussed methodology, what can we do, and what methodologies can we combine. And one thing that we always wanted to do that we already knew how to do is what you see in the first two pictures, which is eDNA collection from flower heads using an ATL buffer. This is really just dipping a sunflower head into a buffer, collecting DNA, analyzing it from barcoding. Then we had preliminary tests in 2024 between Serbia and Austria, where we also tested vein traps. And we did the morphological analysis of all the arthropods we collected in two different sunflower lines. I will show you those results later, and those results are also the reason why we changed the approach for the HelEx field sampling in 2025, where we used tripods, as you can see on the bottom right, with 3 different pan-trap systems in blue, yellow, and white UV colors. And we filled them with propylene glycol to basically fulfill the same purpose as the vein trap, but just different colorations and a more open system to catch more diversity. We did this in 2025 in 3 locations for our research partners in Germany, where the life sits right now, in Serbia, and in France, in Toulouse, at 3 different blooming events, early, full, and late bloom, including pitfall sampling underneath the tripod, and of course 4 controls on all 5 lines of Helianthus in all 3 countries. So you can calculate, this ended up being thousands of samples that we're still processing. The insect sampling in the lab is the next step. I will give you here some photos that you have a good overview of what was done with those pan traps. You see colleagues in Serbia setting up the pan trap system and then collecting the insects from it, switching the propylene glycol. We have the equipment in the field, and then we separate it from the glycol, we fill it up with ethanol, we bring it to the lab in Austria, and then on the bottom you can see we put them on petri dishes, we dry them in a incubator, and then we weigh the insect sample so that we can get an idea of how many insects were in what colored trap, and then combine them together to check the weights for each plot in each line, and so on and so on. These are the preliminary results from 2024 sampling. This is the vein trap system, and we have a lot to say about this project, but the main key is we did eDNA and bulk analysis. And using eDNA and bulk would be crushing down all the insects we collected from the vein traps and doing metabarcoding. And we proved over 90% of the detected species that we detected using the morpho analysis. So this gave us really the information, okay, doing eDNA and bulk and morpho analysis is not really necessary. We can stick to eDNA and bulk analysis. You can also see this very well in the MDS plot. We see the separation between the lines, between the plots, and between the analysis systems for different kinds of groups of arthropods, also between Austria and Serbia. I think we can already go to the next slide, because the key is what we did in 2025. This analysis is still ongoing, so now we have eDNA and bulk analysis. I just finished the bulk analysis on Friday, so lab work is waiting for me tomorrow to continue this process before the new year. We had 648 total samples from 3 different countries. And what you can see, we're also now looking at how many insects were in what colored pan trap, in what line, in what country, in what blooming event, what were the weather conditions, and so on. So we have a lot of analysis to do here, but we have also a lot of information to work with, which is quite exciting. And as you can see on the Excel sheet, maybe just go back for 1 second, there's information on the wild bees. So we don't do a morpho analysis of all the insects. That would be years of work. But what we do is we look at Apiformes, the taxon, and we just write down there were 3 in the yellow, 2 in the white, 6 in the blue, and then we just write down it was Apis, Bombus, Halictidae, Collettidae, so that we have a better overview of where do the wild bees go, the most effective pollinators. For example, in France, there are a lot of bumblebees, in Germany, there were a lot of honeybees, and so on, so that we can gather all this information to do a really comprehensive study on what lines attract the pollinators, what color traps attract the pollinators, and did we get a good overview of all pollinators that visited this. And tomorrow we have a meeting on continuing those trials in 2026 and combining them with insect trapping, which the colleagues at INRA in Toulouse are doing, so that we have another system of proving our approach. Yeah, then last slide. Let's get down to the outlook. So we already saw that novel technologies can foster biodiversity assessments across ecosystems and scopes. The diverse sampling approaches are possible while species detection and taxonomic sensitivity is generally increased. And what's most important, the meta-barcoding of sample may foster upscaling of experiments also in agricultural settings. This is a big market. And then what is key, different methods obviously detect different communities, different primers work differently, and so on. So what we really noticed is that the research question, what do we actually want to know, and the planning process, how can we do it, what factors will affect this sampling, and so on, this is really key. You really need to be prepared, constantly work on your protocols, think what can happen, where do you want to go, because otherwise you will end up with tons of information but have no idea what to do with it. And I think especially in HelEx, we're in a very, very good way to really have a comprehensive study. Yeah, I did it quite fast. With the last slide, I want to thank everybody for having me. If you have questions also outside of this workshop, you can reach me anytime. I'll be there to answer your questions. If I can't answer them, I'll forward them to my colleagues. Thank you.
Thank you, Ilya. I have questions for you. You told us that you collected 1,000 and maybe a lot of data. Do you use actually the AI, the artificial intelligence to assist you on analyzing those results, or maybe in designing your experiments?
I don't. I also don't think my colleagues do. My artificial intelligence is my colleague Johanna, who is an expert in data science and who can work with all those samples very, very well. I don't think it's needed for what we do right now. It just takes a lot of time. But also, if we would include more AI, the questions would still come back of, is this correct taxonomy? Does this make sense? So it doesn't really cut down on time. It's better to have 2 people really working on it full time and getting down to the grid, 1 expert on taxonomy, 1 expert on data science, and then just get the results down.
Mm-hmm. And Rolf, I have questions for you now. You told us that you're using AI to dive into data, reports, and grey literature. Do you think that for commercial situations and agricultural services, advisors on the field, AI will change the game? With your mic on. You just need to switch on your...
Yeah, thanks. Well, I think for the advisory services, AI could be a game changer. I'm not sure whether it's close to it within the next years, but AI systems are currently introduced into legal advisory services, so actually used by lawyers to explore regulations and something like that and give advice further on. And I could imagine that on midterm and on the long run, advisory services could be linked with AI systems. But nevertheless, I'm not sure whether these will be the systems or just the databases that are used by Grok or by Claude and so on.
Or specialized interfaces.
Yeah, I think the basic data need to be provided by the specific owners of the data or by the services, and so they will train it on their own. And for the AI systems, it's also an issue about the copyrights for the data that are used and the what's called now the reliability, as well as the well, who owns the data and who could use the information. And essentially in the agricultural system, I could imagine that some data from agricultural production are sensitive, and they may not be disclosed broadly to the open system of the web.
Thank you very much to both of you. And it's time for me to conclude this webinar. Many thanks to our speakers for sharing fascinating results. Many thanks to you two, dear attendees, for being with us today. And for more information, you can please visit HelEx and Booster websites. You'll find news, resources, and the replays of this webinar and the previous ones. To share your comments and impressions on this webinar, please respond to the short satisfaction questionnaire that is about to appear on your screen just now. Thank you very much, and bye-bye.